body microbiota were scaled according to size, then what we would find is that the colon microbiota, the gut microbiota, would fill the entire screen. That it is by far the largest, most diverse, most complex, and most important bacterial community living in the human body. So much so that sometimes scientists, me included, can get a little slippery in our language, and when we use terms like the human microbiota and the gut microbiota interchangeably. It's not technically true, but this is the one that gets the most attention. So when we look at the makeup of, you can see yourself asking a question, well, what does a really healthy, a beneficial gut community of bacteria look like? And so early on, the thought was that there would be one particular set of microbes that would make for a beneficial relationship with their host. And pretty quickly, scientists realized that's not true. Here in the chart labeled Bacterial Diversity, we see that for different individuals along the x-axis, each one of these individuals has healthy digestion and a beneficial relationship with their gut microbiota. But again, at the level of phyla, which is what those different colors represent, at the level of phyla, we see that there is huge variation in between which microbes are present in these different individuals. So we have realized then in this figure that there is no one set of bacteria that are beneficial for everyone. However, what we did find is if you look at not which bacteria are there, but maybe which genes are present, here we see a surprising level of similarity across those same individuals. So it seems like what's important is not to have a particular kind of bacteria present in the gut, but instead you need a particular job done in that population. Or if we were in the valley one more time, then maybe it's not necessary that you have this exact species of pine tree across the floor of the valley. You need some sort of tree so that it'll provide soil retention and help maintain moisture in the soil and those sorts of things. That with someone doing that job, it'll provide for a balanced and healthy human ecosystem. So from data like those and more, it's become undeniable that the microbiota is important for digestive health. But all along, there's been growing evidence that it also impacts neurological health, and possibly even behavior. There have been a number of studies in rodents and even in human beings where the individuals are fed certain bacteria that is thought to be beneficial, and then they measure changes in behavior. Not all of the stepping stones between those two have been fully worked out, and it's those stepping stones that are actually the part that I find personally most interesting. And so to give you some background on the gut-brain axis and how the bacteria in the colon are able to influence the brain, I want to show you a couple of highlights from this recent and very thorough scientific review written by a team of scientists that include the first author there, John Crine, who is undeniably the world leader in this field. This review covers essentially anything you would possibly want to know about the gut-brain axis. If it is known, those of you who may look carefully at the reference would notice that it's some 150 pages long. It's a worthwhile reading for those of you who have an interest. So to show just one figure from this massive paper, this tries to illustrate some of the ways that it's thought the bacteria in the colon are able to influence brain activity and even behavior. And so I'm going to break these down into a couple of different categories. The first, starting over on one side, is that it's known that the gut microbiota are able to produce neurotransmitters and neuroactive metabolites that can then leave the digestive system, make its way into the circulatory system, and be passed into the brain. In mouse models, rodents with or without a gut microbiota will show changes in levels of neurotransmitters or other important metabolites. So it's thought that maybe this is one way that the gut microbiota can influence brain activity. Another category of molecules, the short chain that are made by the microbiota, are short chain fatty acids, for example, butyrate, which as a group are known to have influence on brain behavior and gene expression in the brain, and a large percentage of the total concentration of these in the bloodstream comes from these gut microbiota. And then the third type of molecules that are made, you see there are tryptophan metabolites. Tryptophan metabolites are able to pass the blood-brain barrier, and many of them are actually produced by resident microbes of the large intestine. So one category of communication is through small molecules made by or modified by bacteria in the gut that pass through the circulatory system and work their way to the brain. A second level of communication happens through interactions in the immune system. Immune cells are present in huge abundance directly against the large intestine. And those cells are also able to work with the brain. So thought is that changes in activity of the immune cells may lead then to changes in brain activity. But a third category of a third mechanism by which communication may occur is through the vagus nerve. The vagus nerve is primarily thought of as a nerve that from the brain controls the activity of the large intestine. Any of you who have ever been in a stressful situation or maybe had a big presentation or something coming up and sensed some gastrointestinal symptoms of that stress have experienced the activity of the brain working through the vagus nerve into the large intestine. What's only more recently becoming known is that the communication also goes the other way. There was an elegant set of studies that were done in rodents where the vagus nerve was cut. And what they found then was that changes in the gut microbiota had a reduced effect, a reduced ability to change gene activity in the brain. It seems as though at least some level of communication works its way up the vagus nerve to the brain. And so with those kinds of information about how the brain and the microbiota interact, studies abound where individuals, either humans or animal models, are fed bacteria and then try to find out what's happening inside their brain. Here's a representative paper like that, where it's looking at brain signatures in brain activity and self-reported emotional response of individuals who are fed either or are given no intervention, which are the blue bars, given a placebo, the orange bars, or fed a beneficial bacteria, which are the green bars. And so what you're seeing is that in the figure labeled A there, is that these individuals, after a few weeks of feeding, report less of a sense of hopelessness, and they have less risk aversion after being fed this probiotic, and that those numbers seem to be statistically significant. This particular study is so interesting because the researchers were able to use these groups, each of which had 15 individuals in them, and actually map onto the brain regions of the brain that are involved in these various activities like risk aversion or hopelessness. And they did see measurable changes in brain activity in each of these. So to me, what I find exciting in this is that it shows that the bacteria can change brain chemistry, brain activity, and behavior, but it doesn't explain the stepping stones in between. Another study that caught my eye has to do with treating children who have autism spectrum disorders, not a probiotic, but a prebiotic. A word on prebiotics. These are an interesting idea, where instead of feeding individuals bacteria directly, instead they're fed complex sugars like we might find in soluble fibers, in hopes that those beneficial sugars and complex sugars will enrich the community of healthy bacteria in the gut in a somewhat more natural way. This is not as risky of an intervention. Some of these prebiotics are found in things like fruits and vegetables. And so in this study, caught my eye not only because I have some interest in autism spectrum disorders, but also because what they did afterwards, after feeding the children the prebiotics, is it then measured for different beneficial gut microbiota, and they measured the levels of different fecal metabolites. And so I highlighted one line on this chart inside a blue box, because the bacteria that line represents is the same one I use in my studies, Bifidobacteria longnum. And they found that changes in this bacteria led to changes in a whole bunch of important amino acids. So here we're getting a little hint that this prebiotic can change for the presence of this bacteria, which may change for these gut metabolites. And so as it's interesting, it's difficult to get to real causal mechanistic explanations in human beings because of the limits of human research. However, in animal models, we're able to do a little bit more. And so this is a remarkable study that was published years ago now in the journal Cell. And what happens here is they take mice and treat them with a treatment that induces autism-like behaviors and brain function in the mice. And so in the bars that you see there, the S is just saline, so that is a placebo or control treatment. And then the P represents their treatment. And so what they find, what that graph is measuring in C, is marble burying behavior. This is a repetitive behavior that represents the kind of repetitive behaviors that are common in some individuals with forms of autism spectrum disorders. And so what they can see is that this marble-bearing behavior increases in the mice that are treated to have autism-like disorders. That's not a terribly surprising result. But what's exciting is when they then give these same mice a beneficial bacteria, a probiotic bacteria, enough to populate the gut, that behavior goes almost all the way back down to normal. And so here again, they're feeding the bacteria, changing the gut microbiota, and then finding a change in behavior. But they were able to go further. They then went and looked at blood metabolites in these mice. And one that is particularly interesting is called 4-EPS, 4-ethyl phenylsulfate, shown here. This is a molecule that it and some of its related molecules are thought to be neurotoxic, and therefore are normally only present at incredibly low levels in healthy mice, as shown in the graph marked A there. Very low levels of this potentially neurotoxic molecule are around in the blood. However, in the mice that are displaying autism-like behaviors, we see a very high concentration of this 4-EPS in the blood. And again, the presence of the blood metabolite is again reduced if the mice are fed this beneficial bacteria. So I think this is a very exciting result because it starts to fill in the space between adding bacteria and seeing a behavioral change that now we're starting to see that maybe this blood metabolite 4-EPS is being affected by the presence of the bacteria, and that leads to some of these brain changes. And so I hope I've convinced you that the evidence for behavioral changes in response to the probiotics is pretty strong in rodents, and that there are hints that correlative changes in human behaviors are also likely true, and more results like that keep coming in. But there's still so much to be discovered. What exactly is the mechanism of the communication? What neurological changes are happening? How are those changes happening? What changes in gene expression are happening? How does that lead to the behavioral changes? This and so much more. So much science to do. And so I was explaining some of these findings in my microbiology course, and a then young student named Kara Horseman, shown by herself here, was sitting in the front row. Kara was a biology, psychology double major, and in the description I was just giving to you, she heard all of her interests colliding. So she ran to me after class and said, please, can we do research on this? Please, please, please. And I was pretty quickly convinced that she was going to do this research, whether I helped her or not. And so I decided, I think we can go ahead and give this a try. I did some background reading, figured out what skills that I already knew from molecular biology and microbiology, what resources did I have on campus that we could pull together and try a project in this area. I then convinced her this was too much work for her to do by herself. Undaunted, she rapidly put together a team by recruiting these other two students to join us, Marie and Laura. And together these students put in what adds up to almost 8 years of work on the project. And so we are now putting the final touches on the paper, and hopefully we can publish. Cross our fingers. So the hypothesis for our work was this, that changes in the gut community, the gut microbiota, will cause changes in gene expression in the brain. And at least in part, those changes occur because of microRNAs, miRNAs. So while I'm sure that most of you are familiar with microRNAs, just to make sure we're all on the same page, I'll say a couple of words about them. MicroRNAs are a kind of regulatory RNA. They exist all over the genome, sometimes as independent genes, sometimes inside introns of other genes. And microRNAs have a whole parallel process of transcription and processing like you might have heard of for mRNAs, the way they get a poly A tail and a cap and all of that. Well, after these microRNAs are matured, they are partnered up with a number of regulatory proteins, and then each microRNA becomes a mRNA repressor. They sometimes will target that mRNA to a certain region of the cell. Sometimes they will stop its translation so that translation of that particular mRNA comes to an end. Or other times, it'll actually destroy the mRNA, which will have the consequence of essentially deleting that gene from the cell. But either way, typically, microRNAs will be repressors of some other gene's expression. So the more the microRNA is present, the less its target mRNAs will be inside the cell. So the beneficial bacteria that we chose to go after, again, being aware that there's probably not one exact bacteria that is helpful, but maybe instead a bacteria that can do a job that wasn't being done that we're looking for. The bacteria we chose is shown here in a gram stain is Bifidobacteria longnum. This is a gram-positive bacteria. It's known to be among the first microbes to colonize the gut of human infants. It has anti-inflammatory properties, and in general, paper after paper say that this is a beneficial bacteria in the human gut microbiota. So we're going to use this, and the idea was take a relatively modest number of mice and then feed them the Bifidobacteria. But of course, if we're going to do that, we need something to compare them to. And so we also have 4 mice who are just going to be fed media as a control. But then we couldn't help ourselves. And so we also repeated the whole thing with another strain of mice called bulb C mice. These are another standard lab bacteria, or I'm sorry, lab mouse. However, they have an important difference in that bulb C mice often have like built-in social problems. So the idea was that maybe this could be kind of like the social problems that are observed in some individuals with autism spectrum disorders. And so if we could compare how the wild-type mice respond to the Bifidobacterium to how the bulb C mice respond to the Bifidobacterium. This is our experiment. So a word on our experimental design. The mice were fed 1 billion Bifidobacteria longum a day, each day for 3 weeks. We started off with 3-week-old mice, and then they spent half their life being fed the beneficial bacteria or control. Afterwards, the brains were harvested, and we did whole brain isolation. And then from there, we withdrew an RNA, which in a way that enriched for microRNAs over other RNAs. Then we did some reverse transcription to make cDNA copies of all the RNA we isolated. And then we used this useful tool that I like a lot called Pcr Arrays from the company QIAGEN. These allow us to explore 80 different neurological or neurodevelopmentally related microRNAs at the same time to measure their expression in each one of these samples. And so the kinds of questions that allows us to ask are these. For a wild-type mouse, fed Bifidobacteria longum, do they show an increase in the microRNA gene expression for all 80 of these microRNAs? Or does it show a decrease? And likewise for the bulb C mice, we can ask, once fed the Bifidobacteria longum, does that lead to an increase or a decrease in any of these different microRNAs? And so looking at this heat map, where you can see that the various colors, green or red, or an increase or a decrease, just stepping back and glancing at it, you can see two things. Do the Bifidobacteria seem to cause changes in gene expression? Yes. And do the two mice respond differently? Yes. And so that was already an exciting result for us. But to try to put a little finer point on it, here we're looking at the expression of all of these different microRNAs from the whole brains of the bulb C mice versus the wild-type mice. This is both without treatment, trying to see these two mice that behave so differently. Do they have any differences in the microRNAs in their brains? Well, since these are both healthy young mice that are the same age living in the same environment, it's maybe not terribly surprising that in general, their microRNAs seem to be expressed about the same amount in both strains of mice. That's why most of these dots center around the center line. However, there seem to be a number of microRNAs that are expressed to a higher degree in one strain of mice versus the other. Those are the ones highlighted in gray. And wouldn't you know it, that the microRNA that's most different between these two, the one that I put a little gray box around, is a microRNA called microRNA 652 3P. And this was a great result, because it turns out microRNA 652 3P is associated with autism spectrum disorders. So we thought, that's a microRNA we need to keep an eye on. So what happens? Well, when the mice are fed Bifidobacteria longum, we see that in general, the two strains of mice are now more similar with each other than they were without feeding. And that includes Bifidobacteria longum, which is now squished to somewhere there in the middle of the pack. This is just the kind of result that we were hoping for. But because we were so excited about it, we wanted to make sure it was really true. And so instead of just using this PCR array, we found a specific set of primers for microRNA 652 3P. We wanted to investigate this exact microRNA, as opposed to the whole population, in the different treatment groups that we had. So here are those data. In the first two bars, we're seeing the control. And so the very first bar, the lightest color one, those are the wild-type mice that are just being fed media. That shows you there is their level of expression of this particular microRNA. Whereas the bulb C mice seem to have a much higher expression of this microRNA. And so there's more of that repressor around inside the cell. And then there, as you can see, is that after feeding, the mice that have been given the Bifidobacteria longum, now we see essentially no difference whatsoever, at least with an error, between the wild type and the bulb C mice, that big difference in gene expression of this microRNA is now completely lost. And so this is the kind of result that we were expecting, that we were hoping for. But in order to try to fill in the stepping stones of how does the bacteria change behavior, we needed to see, does this difference in microRNA level do anything? And so we asked, well, what are the targets of microRNA 652 3P? Are there any mRNA targets that it regulates that could be of interest to us? And sure enough, there was. It's an mRNA called DAB1 that is involved in brain structure. And so we decided, let's measure DAB1 gene expression in each of these different mice in hopes that as the repressor goes down, perhaps DAB1 will go up. And it did. And so here we're seeing that the wild type basically had a fairly low expression of DAB1 with and without feeding. However, there is an increase in the expression of DAB1 in the mice that have been, the bulb C mice that have been fed the beneficial bacteria. That it appears that perhaps as that repressor expression goes down, the presence of the mRNA goes up. So this is a fun result. And so from it, we can draw a few conclusions, like probiotics do seem to influence the microRNA expression in the brain. And changes in microRNA could possibly help explain some of the changes that are observed in the mRNA. And it appears that there are different responses to the probiotic in these two different strains of mice. And so we're glad to be able to make a contribution to this field, but there's still so many unanswered questions. Questions like, well, how much of that mRNA change is really explained by the microRNA? Maybe it's just changes in transcription factors. Maybe it's changes in, well, who knows what. And then we see this change in DAB1 expression, but does that change explain any possible change in the morphology of the brains of these two mice? What if we gave them more bacteria or a longer exposure? What if they started even earlier in life instead of waiting until they're a few weeks old? What about microRNA levels and behavior? Could we actually predict the behavior based on the microRNA level expressions? That's really interesting. And so we tried that one. We tried measuring through classic psychological experiments in mice. Do they have social avoidance, or they are more social than with treatment? And what we found is what has been found in psychology labs around the world is that if you're going to do psychology studies on mice, you need a very large population of mice to get statistically significant data. And unfortunately, we didn't have that. So far, we're not sure if it correlates behavior or yet. Those continue to be work yet to be finished. And so I think that labs are able to collaborate to try to find about how these bacteria in the gut can interact and speak to the brain, causing changes in the brain that eventually cause changes in behavior. The effects of the gut-brain axis in the human microbiota is a vastly exciting field with new results coming out every day. It's difficult to keep up with, but this is exciting work. We're glad to be able to work in it. So I have some references for you here. I can certainly come back to the slide in a moment if you would like me to, but I highlight again that first one, which is that 150-page review that covers everything you need to know. So with that, I'd be happy to answer any questions that you might have and about Trinity, about the microbiota, about our research, or whatever. But again, I'm thankful that you got up this morning and spent some time with me. Thank you so much for your attention.
Thank you, Dr. Clay. Thank you very much for your thought-provoking session. Now the session is open for discussion. Dear participants, you can put your questions in the chat box, or you can ask questions directly to the resource person. Welcome to all the participants to the discussion session.
Hello.
Hello.
Good evening, sir. Sir, you chose this bit of bacterium longum, for your work. We have in our gut vast a variety of microbes are. We have a lot of microbacteria, no?
Mm-hmm.
So how you choose the particular bacterium in your mice variety? How you negate the possibility of other bacterium?
Yep. So thank you for that question. I appreciate it. There are at least 3 reasons that drove us to that question. One was that we had seen other papers that had shown the beginning and the end, that feeding with Bifidobacteria longum would change the behavior. So we thought as long as someone already knew the ends, maybe we could help fill in the stepping stones in between. Second reason was we could get it. We found a place where it was available for sale at a price that we could afford. So we decided we were willing to buy it and grow up some more. So that was just a very practical reason. And the third reason was so we had at least some comfort in the idea that it may not be that we have to find the exact right bacteria. Again, from that idea of the diversity of microbes, that it may not be that there's a magic bacteria out there, but maybe we can find a bacteria that would work in these mice.
Thank you, sir.
Thank you. Thank you for asking.
Any other questions? Participants, please ask questions.
As you see, if you do come up with questions later, you're welcome to email me. I'd be happy to continue the conversation.
Okay, sir. Okay, sir, one question is in the chat box. Shall I ask?
I see it.
Yeah. To what extent does antibiotics affect colon bacteria and brain cells?
Yes, so this is a very interesting question. I think right away we have to point out that antibiotics are a redemptive gift from God for which we are all grateful. I'm sure that many of us here in this call wouldn't be here alive today without the use of these antibiotics. They're a wonderful gift. That said, any medicine strong enough to have an effect is strong enough to have a side effect. And so antibiotics are a powerful and wonderful medicine that can save our lives, but they can also cause vast disruption to our microbiota. The idea of antibiotic-associated diarrhea, which is a common side effect, is a testimony to that truth. Taking antibiotics will disrupt the gut microbiota. The good news is that in most individuals, the gut microbiota, like all other rich, diverse, well-established ecosystems, is resilient, which means that even after a catastrophic event like a dose of antibiotics that hugely disrupts the microbiota there, the overall ecosystem is such that given enough time, it's likely to bounce back to what it was beforehand. The same is true for other disruptions. When you travel, when we're allowed to all travel again, when you travel and start eating different foods, living in a different environment, it'll cause big changes in your gut microbiota. But once you come home and go back to your regular diet, it's likely to bounce right back. Where things are most interesting is for doses of antibiotics for the very young, for babies. And so for those individuals, they do not yet have a well-established microbiota. And there is at least some concern that multiple doses of antibiotics very early in life could be disruptive in a more permanent sense to the bacteria that form in the colon. That said, there's a fair chance those antibiotics are saving the child's life, for which we are all grateful. But again, any medicine strong enough to have an effect, strong enough to have a side effect.
Okay, sir. One more question. Do these affect the genes? If yes, what is the possibility of the genetic evolution?
Yes. So we were primarily just looking at the gene expression. And so we weren't looking at the actual changes in the gene level. There are some new data that show that there's possibility that mutations are higher in the brain than has long been expected. And so there may already be, since there's that change in gene expression there, or a change in gene sequences, there's a possibility that would have selectable phenotypes and therefore affect evolution. But at this level, that's not what we were looking at. We're just looking at the gene usage. Now, more broadly, of course, there's lots to be had here from evolution. That really, a human being, we don't even evolve on our own, but instead we evolve as a holo organism, where we and our broad collection of microbiota evolve together. And whatever is best for all of us is what gets passed on to the next generation.
Any more questions? One more question. Can humans share genes with the bacteria?
Ooh, so horizontal gene transfer between bacteria and human beings. I think probably 10 years ago, the answer is no. And then more recently, it's probably maybe a little. Maybe it doesn't. Yeah, I think the jury is still out on that one, because there's a lot of people in the world, and there's been a lot of people in human history, and we do know that the bacteria in our guts carry around and give to us 150 times more genes than you received from your mom and dad. And so we have this vast genetic diversity living inside of our bodies. And so do we think that maybe any of that DNA jumps from one species to another? Just because the DNA got into our epithelium lining our colon doesn't mean it makes its way into the gene pool and is passed down generation to generation. But in another way, like in the holo enzyme, or the holo organism I was just talking about, it's almost like it doesn't have to, because since we pass on the microbiota across generations, and that absolutely happens, that we pass on that entire genetic contribution from mother to child, through family members, without having to incorporate it into our DNA. It's like the rest of the genetic inheritance we provide is provided through the act of birth, through skin-to-skin contact, through nursing, and so on.
How much time it will take back to flourish our bacteria back to our body after intake of antibiotics after an infection?
For any individual, that's a little bit difficult to say, but in general, in a week or two, you'll be fine. And this is both good and bad, that resilience thing. The good news is you can take antibiotics and it'll bounce right back. The bad news is, if you do pay for and purchase a probiotic and take it, any benefit you get will only last as long as you take that probiotic, because of resilience. Even if you took 1 billion a day, every day for a month, that once you stop, it's going to go right back to what it was beforehand. And so kind of the interesting question we weren't asked is, well, then how do I shape my microbiota? How do I select for a healthy microbiota? And the answer is the same one your parents told you. Eat your vegetables. If you eat your vegetables, that will give a more diverse microbiota and healthier microbiota.
Development of brain. So these again the bifidobacterium infants and diamond infants. So these appears only on the early developmental stage of throughout lifetime.
The question is, as you explained, that Bifidobacteria luminogum in infants miRNA appears only on the early developmental stage of thought throughout lifetime.
Yep. So those results are known. If I remember correctly, I believe DAB1 is expressed in early development and then tapers off. I think that that's correct. And so maybe the reason why we saw it at all is because we started with these 3-week-old mice that were still fairly young. I think if we repeated the results with geriatric mice, we might get a very different result, which, by the way, the microbiota of aging is another fascinating topic, because there are all sorts of microbial changes that happen during aging that might actually explain some of the symptoms of getting older.
Sir, whether future studies are possible in the same field of your work?
Yeah, blood pressure and gut microbiota. Actually, this is, I'm so glad somebody asked this question. The really interesting result with blood pressure, that is what you asked, right? Yes, blood pressure and gut microbiota isn't the gut microbiota. It's actually the oral microbiota. And so there are species of bacteria that live in the mouth, and particularly on the tongue, that produce a lot of our nitrous oxide, which helps regulate blood pressure. And so there are interesting studies where people who used antiseptic mouthwashes or did tongue scraping, which is common in some cultures, in addition to brushing teeth, would see a spike in blood pressure afterwards and a change in levels of nitrous oxide. But then as that oral microbiota regrows throughout the day, because of resiliency, that as it came back, the nitrous oxide levels would stable off. That's a very interesting study. And so if blood pressure issues run in your family, quit the mouthwash.
Another question, how probiotic bacteria affect the neurobiology of the animal? Probiotic bacteria.
I see that one now. How can probiotic bacteria affect the neurobiology of the animal? I don't know if I can answer that question effectively. I think I'm at the level to say that it can affect the neurobiology. That was already an exciting enough result for me. I think how, it's going to be too complicated for me to answer with which species of animal and which species of bacteria and in what age group and how and so on. Still more work. Jump in on it.
Please explain the relationship between high blood pressure and gut microbiome.
Yep, and so I addressed that one. That's the one with the oral microbiome and tongue scraping and antiseptic mouthwashes.
Medicine for coronavirus? Okay, sir. One more question. Effects of food additives on gut microbiota.
By and large, if we eat it, they will eat it. And so there's been a million studies where you feed someone molecule A and see, does it change their gut microbiota? And almost everything does to some extent. And so the question is, just because it causes a change doesn't mean we know if it's good or bad, because we don't yet know what a good gut microbiota is. And so can it change? Yes, but is that a problem? We don't know.
Dear participants, please mute your microphone. If you are asking questions, you can put on the mic. Otherwise, please mute the mic. Disturbance coming. Please. It's a really pleasure if you are sharing your email. Okay, email is already provided. Take the email from the
Email me, Miera. I'll send you something.
Sir, can you please share some reference which could explain blood pressure and gut microbiome? That's I think already.
Yep. Send me that email in a question. Send me that question in an email, and I'll send you back a link.
Okay, sir, I think almost all questions are finished. Thank you for the discussion part. Thank you, sir. Now I welcome Anjali Rajendran, student coordinator, to propose the vote of thanks. Anjali.
Good morning, one and all. I deem it a great honor to propose a vote of thanks to all who have helped us in making this webinar such a resounding success. First of all, I would like to thank our resource person, Dr. Carlson, Professor of Biology, Trinity Christian College, USA, who spared the time from his busy schedule to grace this webinar with extremely relevant address on genes, bacteria, and miRNA, beneficial bacteria, and the gut-brain microbiota axis. Thank you so much, sir, for sharing your research experience and giving us an idea on how the gut bacteria influence the brain, the response it produces, and also for clearing our doubts and making the session more lively. Once again, thank you, sir, for such an informative session. We have been fortunate to be guided by our principal, Dr. Sister Beena Jose. You have been a constant source of guidance and encouragement. Thank you so much, sister. Next, I thank our Vice Principal, Dr. Sister Namita, and Dr. Minimon Ma'am for their enthusiastic support and guidance. I thank our well-wishers, Dr. Honey Sebastian Ma'am, Head of Department of Zoology, and Dr. Shijati Tharigan Ma'am, Head of Department of Botany, for their support, vision, and commitment. Events like this cannot happen overnight. The wheels start rolling weeks ago. It requires planning and a bird's eye for detail. I take this opportunity to thank our teacher coordinators, Dr. Sister Sylvia Matthew and Dr. Honey Sebastian Ma'am, Organizing Committee, Dr. Shijati Tergan Ma'am, Smita P.S. Ma'am, Dr. Manju Madhavan Ma'am, Dr. Patricia Joseph Ma'am, Dr. Fibarani John Ma'am, Dr. Shiva P. Ma'am, Sister Frenny Jacob. I thank student coordinators Hira Babu, Helen Nixon, and Sandra Essen, who have been running around to make this webinar a grand success. We are really grateful to be funded by DBT Star College Scheme, Department of Biotechnology, Ministry of Science and Technology, Government of India. Finally, I thank the wonderful participants who turned up in such a great number from various departments. Thank you so much for your cooperation. Once again, thanking each and every one of you. Thank you.
Thank you, Anjali. All my dear teachers, please switch on your videos for taking a photograph of the session. Please switch on the videos. Thank you all. Thank you once again. Thank you, sir. Thank you. Thank you. Now let's leave the meeting. Thank you.